Hey, everyone, Jyoti here. You know how much I love genetics, and today we're diving into a fundamental process called gene annotation. It's like finding treasure hidden within our DNA. Imagine having a whole library of books written in a language you don't understand. That's kind of what our DNA is like. Gene annotation is how we decipher those books, figure out what each gene does, and how it contributes to the incredible complexity of life. In this video, we'll be your guides on this journey. We'll break down the essential steps to annotate a gene sequence using some awesome online tools called genome browsers. Get ready to explore the fascinating world of genes and unlock the secrets hidden within our DNA. Our first step is choosing a genome browser. Think of it like picking the right map before you set off on an adventure. There are a bunch of great options out there, each with its own strengths. Some popular choices include the UCSC Genome Browser Ensemble and NCBI's Genome Data Viewer. Each browser has its own unique layout and features, but they all essentially do the same thing: visualize and annotate genomes. Don't get overwhelmed by all the options. We'll link to some helpful resources in the description that compare these browsers and their features. For this walkthrough, we'll be using the UCSC Genome Browser since it's super user-friendly and has a ton of cool features. But seriously, explore different browsers and find the one that feels most intuitive for you. It's all about what helps you navigate the genome most effectively. Alright, we've got our trusty genome browser open and ready to go. Now it's time to find the specific gene we're interested in. Think of it like using the search bar in your favorite online library. We can either type in the gene's name, like BRCA1, or its unique ID called an accession number, which is like a book's call number. Let's say we're curious about the BRCA1 gene, which is involved in DNA repair and linked to certain cancers. We'll type BRCA1 into the search bar and hit enter. Boom! The browser takes us directly to the location of BRCA1 within the vast landscape of the human genome. See how easy that was? Now we're in the right spot, ready to start digging into the details of this fascinating gene. Now that we've found our gene, let's zoom in and get a closer look at its structure. Remember, genes aren't just long, continuous strings of DNA. They're organized into sections called exons and introns. Think of exons as the important coding sequences that get translated into proteins, and introns as the non-coding regions in between. The genome browser lets us visualize this structure in a clear and colorful way. We can see the exons and introns laid out, along with other important features like the gene's promoter region, which acts like an on-off switch for gene expression. We can even see different transcript isoforms, which are like different versions of the gene that can be produced through alternative splicing. It's like we're looking at a blueprint of the gene, understanding how it's put together and how it might function. We've got the gene's basic structure down, but now it's time to delve deeper into its functional elements. These are the specific regions of the gene that play a crucial role in its activity. We're talking about things like coding sequences, which provide the instructions for building proteins, and untranslated regions, or UTRs, which regulate how, when, and where a protein is made. The genome browser is our best friend here, providing annotations that highlight these functional elements. We can see where the coding regions are, pinpoint the locations of UTRs, and even identify regulatory elements like promoters and enhancers, which control gene expression. It's like we're uncovering the hidden machinery within the gene, understanding how each part contributes to its overall function. Our final step is to see how our gene has stood the test of evolutionary time. We do this by looking at conservation tracks, which show us which parts of the gene are similar across different species. Think about it. If a particular region of a gene has remained relatively unchanged throughout millions of years of evolution, it's probably pretty important. The genome browser lets us compare our gene with its counterparts in other organisms, highlighting regions of high conservation. This information can provide valuable clues about the functional significance of different gene regions. It's like we're tracing the gene's history through the tree of life, gaining a deeper appreciation for its role in shaping biodiversity. And there you have it. We've just walked through the essential steps of gene annotation, from choosing the right genome browser to analyzing evolutionary conservation. Remember, this is just the beginning of your journey into the world of genomics. By using these powerful tools and approaches, we can unlock the secrets hidden within our DNA, understand how genes function, and gain insights into the complexities of life itself. So go forth, explore different genes, and see what amazing discoveries you can make. Don't forget to subscribe and hit that notification bell for more genetics adventures. Happy annotating!